And the next speaker is Tiedong. He will talk about coarse-grained modeling and simulation of DNA condensation induced by multivalent cations.
So good afternoon, everyone. Thank you for the introduction, and I'm really honored to be here to talk about our modeling study of DNA condensation induced by multivalent cations. So first off, why DNA condensation? Of course, as we all know, DNA condensation can be regarded as a potential model for DNA compaction in, for example, eukaryotic cells. And another interesting thing is in various capsules, it's more like a DNA contact with DNA instead of DNA protein complex situation, such as the situation in eukaryotic cells. So DNA condensation here is more relevant. Lastly, DNA condensation, someone just proposed that if we want to do gene therapy by delivering DNA into some cells, then we have to know how DNA condensates and how to modulate DNA condensates. Then we can just do the delivery more smoothly. And DNA condensation is not a new topic. This picture is this cryo-TEM picture is actually from more than 40 years ago. It actually showed a DNA condensate in a very dilute solution. So as you can see, in the dilute solution, if we add in some condensing agent, DNA can collapse and form this kind of a toroidal structure. And the size of this kind of particle is around 100 nanometers. More higher resolution EM picture shows that within this toroidal structure, DNA molecules are organized very orderly. So DNA molecules can be seen as compacted in a hexagonal arrangement. And of course, as you can imagine, the electrostatic interactions are really crucial here because we have to overcome the negative charge repulsion from the DNA molecule. Our goal is to simulate this kind of structure and see what kind of property we can deduce and what we can learn from it. But how do we simulate this thing? It's really big. Atomic simulation would not be a very good idea because the size of this is around hundreds of nanometers. So we go to the coarse-grained route. We use the method of structure-based coarse-graining, specifically inverse Monte Carlo, which is introduced by Professor Lubashev just now. So I'm not going through all the theories and really basic stuff. I'm just walk you through the practical steps of how do you apply this inverse Monte Carlo method to your system of interest, and how to build your coarse-grained model and run the coarse-grained simulation. So the goal of structure-based coarse-graining method is just to get effective potential that can reproduce the properties of your system, specifically structural properties. So first of all, you should have a reference system. More likely, it's an Oitum model, which is probably the most widely used model right now. And then you have to define your mapping rule. So in this way, in this step, you can just remove all unimportant degree freedom. And once you have your coarse-grained representation of your reference system, then you can calculate the structural features of your system, of course, which are just radial distribution functions. And from now on, you are into iterative procedure, whereby you have to have an initial guess of your so-called effective potential. Then you will have a simulated structural feature of your system, represented by the green curve here. Then, of course, it's different from the structure you want, which is presented by your reference system. Then, based on the difference between these two RDF, you can derive the potential update. Then you apply to your original potential. Then you have a new set of potential. Then, of course, you iterate. Hopefully, it will not take you a lot of iterations. But generally, in your experience, you would have to do at least 15 iterations. And then, congratulations, you just reached the convergence, meaning the potential doesn't change very much from one iteration to another iteration. And the structural features, which are the radial distribution function you get, is virtually the same as from your reference system. And for more details, you can go to the poster from my colleagues, poster number 13 and 14. OK, so let's go to our DNA models. We actually derived the two DNA models at the two different levels. The first model is a coarse-grained model. We just call it CG model. Then we try to map every two base pairs to 5 Bs. Actually, this is based on a previous model, but OK. Anyway, and as you can see, at the center we have bead representing nucleosides, and along the backbone of DNA we have two kind of polyelectrolyte chains representing the phosphate groups. And we have four bond types and 3 angle types. Together, we just have this coarse-grained DNA model. We have no charge at the central bead of DNA, but we do have charge on these phosphate groups. And to use this model, actually this model was designed for simulation together with explicit ions and implicit water. OK, then based on this model, we go to another level of our coarse-grained model, which is just a bead-on-stream model. Because this is another higher level, we call it a super coarse-grained model, which is just SCG. It's really simple. It has only one type of beads, then one angle type and one bond type. Here, we even make the electrostatic interactions implicit, meaning there's no charge on these beads, and so that we can go to a very long time scale and a large spatial scale. So let's look at the simulations we have conducted. First of all, we have a reference atomistic MD simulation, which is in a qubit box, and we put 4 double-strand DNA inside. Then in the presence of cobalt hexamine, which is our condensing agent, we just simulate this system to microsecond time scale. Then we use these trajectories as our starting point to build our coarse-grained model. And the next, we go through the MC procedure. Then we have our CG model. And we run our coarse-grained simulation with the CG model at around 1,000 times bigger box. And as you can see these brownish particles, which are DNAs after condensation. And based on these simulations, we go one level higher through the inverse Monte Carlo procedure. This time, we even don't have ions. By the way, these blue and green dots are ions in our simulation. And for the higher SCG model, we even don't have any explicit ions, and we don't have any charge. So we can use really large box and put a lot of DNA inside, then just simulate for long effective time length. Okay, so for the following of my presentation, I will try to show basically 3 simulations. One based on the CG model, the other 2 was based on SCG model. So in this first simulation, we put about 200 DNA molecules in a qubit box. We put them randomly at the beginning, and then we put in a suitable amount of cobalt hexamine, our condensing agent, and another other kind of physioelectric condition ions. Then we started this simulation with Langevin dynamics. So as you can see, the energy profiles goes down really quickly, which is indicating DNA molecules are coming to contact with each other. Then this condensing process happens really rapidly. Then after about 17 million steps of simulation, we reach this state where there's no free DNA single DNA molecules. The DNA molecules are all went to these DNA condensate particles. And next, the next simulation was based on the SCG model, which are just the simple B downstream model. So in this time, we can put more DNA inside because it's a lot simpler. Then the energy profile is very similar to the previous simulation, which is rapidly decreasing at the beginning, then reach equilibrium after about 15 million steps. So in this simulation, DNA molecules are just coming together, forming condensates, and actually you can see a lot of condensated DNA particles. And if we look closer, actually these DNA molecules are highly ordered, and they are packed in roughly hexagonal fashion. So this is really encouraging because we actually didn't expect so good result because the model is so simple. It's just B's on string. And comparing to these cryo-TEM images, they are really kind of match. So the last simulation I'm going to talk about is this simulation with longer DNA, because previously we just used DNA of hundreds of base pairs. This time we want to see what happens to a longer DNA. For example, we have a 10 kb DNA here. Then we just build a long DNA, and starting from the extended linear conformation, started this simulation with this linear conformation. Then, interestingly, after some about 20 million steps, we can see this a little loop was formed on one end of the DNA. Then this loop just grow, and it can pull in more and more DNA. And at the end, we have a toroidal condensated particle here. And if you look closer, this particle is, I would say, relatively well organized, very similar to the gravity image we saw, and we can see some kind of DNA crossing in the toroids, which is suspected by other people because you cannot have a perfect kind of wooling of DNA in a toroid. You have to have some kind of a DNA crossing. And then we try to cut this, try to see what would it like if we cut this toroid. So what would the cross-section look like? Then we try to cut it here. Then surprisingly, in the cross-section, the DNA molecules are organized nearly in a hexagonal arrangement. So this is really interesting, even though this colloidal structure is supposed to happen for flexible polymer, but we still are very happy with our results because our model, this model is really very simple. And I'd like to sum up our presented work here by saying that we have built two coarse-grained DNA model at different resolution levels, and the important point is that within these two models, there's no empirical interaction parameters. These interaction parameters are all physically sound, and these simulations showed that our model is pretty good at reproducing the structural features of a DNA condensate, and we hope to apply our model as well as our method to further more complex structures such as nucleosome and chromatin. And with that, I would like to thank our group members in Prof. Last Skills group, as well as Professor Lubasev and our funding support from MOE, and a lot of CPO hours from ICC Singapore. And thank you for all your kind attention, and I will try my best to answer your questions.
Thank you. Time for question? Yes.
Very nice, thank you. I was curious if you looked at the energetics of DNA condensation within your coarse-grained model. You have the energy numbers, right? You start from somewhere where the DNA is not condensed, and then it condenses, and there's a pretty large drop in energy, 5,000 kcal per mole. So I was wondering if you try to divide that number by the number of base pairs you have, and do you get something that's reasonably describes experiment? From what I remember, it's about a fraction of a kT per base pair for condensation. So how well are you doing on the energetics?
I haven't done the division, the energy difference by base pairs, but I can see that in the interaction potential of our beads, the interaction potential between two beads are about, the lowest point comparing to zero potential is about minus 2 kcal per mole. Minus 2 kJ per mole. Then if we divide by, each of our bead represents 6 base pairs. If you divide by that, we can get a rough number of what's the order of magnitude.
Thank you.
Okay, let us thank the speaker once again.
Thank you.